recording this. There's not very many people here. What I'm gonna do is not write the code on the board. Okay, if you want a point for coming today, you can go to socrative.com. I don't have a blackboard marker, but hopefully you can remember how to spell my name, Lazerus, Lazarus 5825. Go log into that room and sign in with your name, and I will give you a point for coming. Good job. I don't know. There's like less than 50% attendance today. So what's the password? I don't know that there is. Do you need one? It's asking. Password is Happy Wednesday. It shouldn't be case sensitive.
Is there a space?
Socrative.com. I'd write it on the board. I threw away the marker because it was useless. Here we go. Maybe this one will work. I can like not read this. I should start carrying around markers. Okay, and once you're done, let me know and we will get going. Yeah, spring break has not started yet. It is only Wednesday. Plus, I'm giving you guys Friday off. There's no reason to be absent today. I am going to be on Friday. Okay, we're going to get started for today. My only reminder for you is that there's no class on Friday. I'm not going to be here, so I'm not going to make you be here. A lot of students have been asking me about the exam date. Based on the way that we've kind of been going through content, also with the snow days, I don't have that exact date set. It's definitely after spring break. It's not like the Monday after spring break. I'll give you notice. I'll look at it over spring break. I have some time to kind of work on stuff. So I'm going to look at that and try to set an exact date so you can be a little bit more focused. Yeah, I can post a study guide. Yeah. Was that your question too, Cade? Okay. Yep. I do, of course, recommend studying as we kind of go along. So each day after class, I post the lecture recording, and then there's also the PowerPoints that I post as well on Blackboard every day. So I do recommend kind of going through those, but I will post a study guide and I will get you an exact exam date. Any other questions? Like I said, don't come here Friday. I will not be here. Last class, we were talking about mitosis, and I do want to finish up mitosis. We did get through metaphase, which is where basically it's like kind of the middle point of the exact splitting. So metaphase is characterized by chromosomes being lined up on the metaphase plate, which you can see in this illustration. The red X's are our chromosomes. During metaphase, they're maximally condensed. So remember that condension is basically facilitated by protein complexes. So there's proteins that are combined with the DNA, and they can move how much they are condensed. So it's variable based on different times in the cellular cycle. So at this point, they have maximal condension. That's beneficial because when they're pulled apart, they'll be protected, which is important because we really don't want our DNA to be damaged at any point. When we're talking about cancer today, that's going to be evident as to why, in part. After metaphase, the proteins that hold the two duplicated sister chromatids together are going to be cleaved, essentially, and that's when the sister chromatids break apart and they once again become chromosomes. So remember, here we have a duplicated chromosome, and then here we have a chromosome. So that confusing vocabulary is still kind of coming here. As I was saying, the sister chromatids separate. Some vocabulary reminders here for you as well. Now, the chromosomes are pulled apart by attached spindles. So pieces of the cytoskeleton are facilitating this process. At the same time, we do still see the cell lengthening. So it's going to become more oval in shape. And you can kind of see this in the fluorescent microscopy image down at the bottom. This is kind of interesting, but here we can see some of the different proteins that are pulling it apart, and we can see our nuclear material as well. In this illustration, we're noticing like the mitochondria, those are present. They're kind of just reminding you that organelles are also being pulled to the different sides or the different poles of this cell. And that's important because when it splits and we get our daughter cells, we're going to have two entirely new cells that should be identical to our original parent cell. The last phase of mitosis is telophase or telophase, and this is when the chromosomes have gotten to each pole or either side of that more oval cell. Basically, the spindles and the other proteins that pulled the chromosomes apart are going to disassemble. They're going to reform the cytoskeleton in the new daughter cells. And then another key feature of telophase is the nuclear envelope reforming. So we were talking last class about kind of what happens to it. Basically, it gets disassembled and then it kind of hangs out until this final phase where it's reassembled around both of the nuclei for those daughter cells. So the parts are used later on. We also have the mitotic spindle breaking down. So the structure that's kind of holding those chromosomes together. At this point, we would say that mitosis is complete because remember, mitosis refers to the nuclear division. The next step in this process is going to be the cytoplasmic split. I did include this image of different stages of mitosis under a microscope. So you can see the blue kind of squigglies, those are representing chromosomes, and all the red bits are kind of like our spindle fibers. Those are our cytoskeleton bits that are facilitating the movement of those chromosomes. I have a practice here for you. So there's 3 questions here. First one, leopard frogs have 26 chromosomes in their somatic cells. This is true. How many sister chromatids will be in a leopard frog's cells at the end of the S phase before mitosis has occurred? Then I want you to also tell me how many chromosomes at that point. Then the next question is how many chromosomes will be in each daughter cell after mitosis? So if you want to talk about this with someone, you're free to, but try to think of the answers to these questions. This is good review. Yeah, and key to thinking about this and getting started at the end of the S phase. So what happens during the S phase? A couple of different answers. So S phase, we can think about representing for synthesis, and that's when DNA is replicated. So at the end of the S phase, we have double the chromosomes, right? We have our duplicated chromosomes. So does that help answer that question? How many sister chromatids? Does anyone agree? Do y'all think 52 sister chromatids? Okay, we're getting that because we have 26 chromosomes, right? So if we duplicate those, then we would have 52 sister chromatids. Okay, how many chromosomes would that be? 26, yeah, so kind of funky. If our chromosomes start like this and we have 26 of them, and then they become duplicated, we have our sister chromatids, but this is still one chromosome. So yes, correct. And then this last question, how many chromosomes will be in each daughter cell after mitosis? Thoughts? 26, yeah, kind of tricky, kind of a trick question. It is also 26, right? So we're starting with 26 and we want to end with 26. Does that make sense to everybody? Okay, I'll bring a marker that works next time. I don't know why this is happening. Next, no, I mean, unless you want me to draw more chromosome drawings, but extra expos. I think we're good for right now, but thank you. After mitosis, we do have that cytoplasmic split. And these slides, I have a lot of text on them. So I'm not going to go over this like a ton. And to be honest, I'm not super duper concerned that you know the exact details of how cytokinesis works in each different type of cell. Mostly, I want you to know that cytokinesis refers specifically to the splitting of the cytoplasm, basically, and that it's different in different cell types. So what this figure is showing is some of the different types that cytokinesis varies in. So we have plant cells, we have animal cells, I have yeast up here. So we can see kind of these differences here. And then I have those laid out specifically on each of these slides. So this is how cytokinesis works in animal cells. And we can see with our animal cells, of course, we only have a plasma membrane, whereas with plant cells, we have a cell wall, which contributes to the different process that we see here. So during late anaphase, basically we get something called a contractile ring, which forms kind of like a cleavage furrow, which is where the cells basically start to kind of pinch apart. That's what we can kind of think of this as. After that cleavage furrow is deepening, because of the structure of phospholipids and because of the way biological membranes want to self-associate, we do see them reforming that membrane, and then we have those two daughter cells. Now we can kind of think of the contractile filaments that are important in this process as like strings, like pulling a string and tightening that cleavage furrow. That's kind of how they work. So I like this figure. It's fairly simple and it gets the point across here. For plant cells, it's a little bit more complicated, again, because we have that cell wall. So not only do we have to separate the plasma membrane or the biological membrane, we also have to reform that cell wall, which is where Golgi vesicles come into play. So interestingly, Golgi vesicles kind of align at the former metaphase plate. So in the middle of that new cell, or in the middle of those two new cells, they're going to fuse, and that's what forms the cell plate, which basically turns into the cell wall. So again, a bit more complicated for plants, but still not too crazy. And this figure, we can kind of see some of the key pieces here are Golgi vesicles forming the plate, and then eventually our cell wall. So I don't want you to spend like too much time thinking about this. Honestly, if you're studying mitosis, it's more important to spend your time focusing on what's kind of happening prior to the cytokinesis. So thinking about that actual nuclear split. But again, I want you to kind of know that plant and animal cells have a different process. And one thing I forgot to mention about the vesicles here, vesicles as a component carry materials. Remember when we were talking about organelles, we kind of briefly mentioned vesicles. They carry the materials inside them in order to form the cell wall. So that's how that process occurs. They have the materials needed inside. Questions here on cytokinesis? Okay. So after mitosis and cytokinesis occurs, the daughter cells are either going to go right into more cell division, or they'll go into something called GO, which is essentially like a temporary resting state. We refer to GO as an inactive stage. And GO is not technically part of the cell cycle. It's going to kind of leave the cell cycle and enter GO. Now, sometimes this is triggered from environmental conditions. So if a cell is in a nutrient-poor environment, it might go into GO because it doesn't have the necessary materials to continue dividing. Oftentimes, this is affected by growth factors, and growth factors are specific proteins that basically regulate the cell cycle. Oftentimes, if a cell is entering GO because of environmental conditions, it might stay there until those conditions improve. And this next slide, I'm talking about some of the specific cells that are going to spend more of their time in GO versus some cells that are going to more regularly divide. So in humans, for example, some tissues are constantly needing new cells. And we could think of those types of tissues as like the lining of the stomach, for example. It's a really acidic environment. It can be relatively harsh. So new cells are needed there fairly frequently. Another example of cells that need to be constantly regenerating are blood cells, like red blood cells. We need lots of those all the time. And if you think about donating blood, the reason that that's not like a super damaging process is because we have the ability to quickly make more blood cells. So we do see blood cells regenerating often. Skin cells are another one, like epidermis cells, also like the lining of your mouth. So again, if you're in lab, remember we were taking cheek cells. Those are easy to get because they're constantly shedding. Same kind of situation, there's digestive enzymes, there's a lot of digestive kind of components. You might like bite your cheek, and if you do that, you can kind of get those tissues repaired in just a couple of days. So cheek cells are going to be constantly kind of dividing. There are some cells that never or very rarely divide. I'm curious what you think might do that. Do you have any thoughts here? What cells are probably not going to be dividing very often? Sure. Brain cells, yeah, perfect. Yeah, neurons, absolutely. Any other thoughts? If you're thinking of like organs that don't grow, for example. Okay, I'll tell you. Cardiac muscle cells are a great example. So, like in your heart, you're not getting new cells very often, if ever. And honestly, most cells in most organs are not usually actively dividing. They're going to typically be in GO. Now, what's kind of cool is certain signals can actually call those cells out of GO and can stimulate that cell division process. So, like liver cells, they can grow and divide, they just don't always do it. So, like liver damage is it's possible to come back from like a damaged liver because they have this capacity to kind of oscillate between these different cell phases. But the reality is most cells are kind of regularly in GO, except for the ones we were talking about here that are going to be frequently dividing. The last thing we need to talk about relative to reproduction and kind of cell division, bacteria, so prokaryotic organisms, they have a very different process. So when we were talking about mitosis, again, that was focusing specifically on eukaryotic organisms with linear chromosomes. Prokaryotic organisms tend to have a single circular chromosome, which is kind of interesting, and it's a defining characteristic of these groups of organisms. And that's what this figure is showing here. We have this like spirally situation, this spaghetti looking thing. That's representing that single circular chromosome. So in bacteria and other prokaryotic organisms, basically what happens is that chromosome begins to replicate. And as it replicates, the cell has to get bigger to accommodate the amount of material inside. It's going to stretch and kind of just break apart. So they're going to kind of just split as a result. Now, the word binary fission actually means division in half. Binary is like two, and fission is like to split. So we could split it in half. Now, this process does have some specific proteins and things that are kind of regulating it, but basically, a key piece to kind of focus on is the origin of replication, which is highlighted in this figure in green. So the origin of replication is exactly what it sounds like. It's where the chromosome is going to begin being replicated. Typically, there's two origins of replication, and they're going to start working at the same time in opposite directions, and they'll meet each other at the end. What's kind of cool is there's a specific protein called green fluorescent protein, and it fluoresces green, particularly under fluorescent microscopy. And scientists were able to kind of discover how chromosomes move in bacteria by tagging the origin of replication with this GFP protein. GFP has been really useful in a lot of different scientific discoveries, and if you're looking for like a weird research topic, GFP is kind of crazy. So, questions on binary fission? Cool. Now I know this seems kind of ancillary, but this is actually really important if we think about like how many prokaryotic organisms exist on the planet. There's a lot of them. So although we don't spend as much time talking about this, it is still super important. Now, your last little break, or kind of the last thing we'll talk about with mitosis today, I want you to review really briefly the cell cycle and the stages of mitosis. I want you to jot down simple drawings of both of these with labels. So do this really quickly before we get into cancer. And if you can, try to do it without your notes. If you have to use your notes, that's okay, but try to do it without. So the cell cycle and then the stages of mitosis. You can use the brains near you. There's 5, but 2 of them start with a P. 2 of them start with a P. Say what? I'm not going to tell you that quite yet. What's the first? What were you saying, Ife?
Now I'm getting nervous.
Sorry, walk away. So prophase is going to be your first one. Yeah, some people remember the stages of mitosis by thinking about PMAT. PPMAT really would be more representative because we have prophase, prometaphase, right?
See,
Gave you the answer.
it's like checkpoint.
No, not yet. After today, maybe, but no. If you want, I guess if you want to, you can. If you want to go the extra mile and include cellular checkpoints, do it. How are we doing here? Should we go over the cell cycle, maybe, to start? Cell cycle? Okay. There's not necessarily like a starting and an end point since the cycle is cyclical, right? So it's kind of ongoing. But what are our main important phases of the cell cycle? If we have two categories to start, one of those categories is mitosis. And what's the other one called? Starts with an I. Interphase. Yep. So remember, mitosis is a small part of the cell cycle, and that is where we have the cells actively dividing, right? And then interphase is everything else. That's when the cell is growing, acquiring the necessary nutrients, replicating its DNA. So we have interphase and mitosis. And then within the interphase, we have G1, right? Our first gap, we have our synthesis. And then what else do we have? G2, yep, which is our second gap. So typically during the G1 and the G2 phase, the cell is kind of growing, acquiring necessary materials, getting energy, etc. And then the S phase is really defined by the DNA replication. Okay, what about stages of mitosis? Do you need more time for that one or did you get it? A little more time? Okay, we'll spend another minute or two here for that so you can finish your simple drawings of mitosis. Say what? There is such thing as interphase. Not in the stages of mitosis. Interphase is everything else besides mitosis. And I do have a figure of this in the next couple slides. So let's really quickly go over mitosis. As I was saying, there's like 5 stages essentially, and a lot of people choose to remember it by PMAT, which represents what are our different stages. Say them in order. Prophase. Okay, cool. Sorry, what was that second one? Prophase. Prometaphase. Cool. Metaphase. Anaphase. Telophase. Cool. Yeah. And like I said, I have a figure in a couple of slides here for that to kind of go over as well. So in your textbook, your online virtual textbook, there's not a ton of information on cancer. That said, it is a pretty important biological topic. There are some textbooks that have more content on it. I have one in my office. If you want to come look at it, you're free to. I can also provide supplemental materials. There's tons of research on cancer. This is a very, very active area of research. To be honest, this is an extremely brief overview. We do have a cancer biologist in our faculty in the department, and we also have a whole class on cancer biology. So if that tells you kind of like the depths within you can get into cancer, there's a lot that you can look into. So by no means, this is not comprehensive. We're just talking kind of briefly about how cancer can occur and what it is. So we're going to talk about what it is, some of the key vocabulary. A really, really important learning objective is understanding cell checkpoints and what causes uncontrolled cell growth. That's probably our most important learning objective for today. We're also going to discuss a little bit cancer risk factors because it can be relevant. So as promised, here is a figure of the cell cycle. So I was showing you this previously. This one is highlighting specific checkpoints, but when you look at this figure, and it's hard for me to tell what the colors look like out there for you, but this outer gray line is representing interphase. So all of this is interphase. And then this little sliver of the pie, that's mitosis. And so within this mitotic phase, that's where prophase, metaphase, anaphase, telophase, cytokine, that's where all that's happening. Does that make sense? So mostly cells are in interphase, growing, accumulating nutrients, synthesizing DNA, etc. So within these cellular phases, they're typically characterized by specific molecular events. And most of the time, moving from one phase to the next is dependent on proteins, other types of molecules, sometimes RNA. We also know that it's regulated by genes. There's other extracellular factors that stimulate cell division and the movement between different phases. We also know cell division is variable within different cell types. So as we were kind of just talking about, some cells divide a lot and other cells do not. Now, going a little bit deeper into the cell cycle regulation, the events for each stage of the cell cycle depend on completion of events in the previous stage. So essentially, a cell is not going to move from G1 into the S phase until certain things have occurred. Sometimes that could be cell size. If a cell is too small, it's not going to be able to pass the checkpoints into the next phase. If DNA replication is incomplete, for example, it won't go into the next phase. A really important checkpoint is when the cell is verifying to ensure that chromosomes are properly attached to the spindle, the mitotic spindle, so that they can be pulled apart. Because if they're not properly attached, obviously we're not going to effectively separate the chromosomes the way they're supposed to be. So that can be a big problem. So events are basically part of what dictate when the cell will move on to the next cycle stage. The cell cycle is triggered and coordinated by molecular mechanisms. In addition to internal and external signals, I've talked about all of these things so far. And a guiding question here, what happens when there's problems with this cell cycle regulation? What happens when this goes wrong? And that answer is a lot of the times cancer is what happens when cell cycle goes wrong. Now, this data is from 2021. I was looking for more updated mortality data, and it's a little bit obscured at the moment, so I was having trouble sourcing more recent data, but although this was just 4 years ago, this is still relatively true, although like the number of deaths might be variable, especially I would expect probably COVID-19 deaths to be certainly lower. But heart disease and cancer are still the main ways that people in the United States tend to die. Now, when we think about heart disease, that's a huge category, right? So heart disease encompasses a lot of different types of illnesses and things. And so does cancer. There can be lots of different types of cancer, but the idea here is essentially problems with cell cycle regulation are a big deal. There's a lot of cancer deaths that occur because of this. Typically, most common cancer types are breast cancer, lung cancer, colorectal cancer. Now, the most predominant type of cancer tends to vary if you're looking at like men or women. It also varies. I don't know why it keeps changing randomly. It also varies based on other like socioeconomic factors and demographic factors. So there is variability in kind of like what types of cancer are the most pervasive. One thing, and I should have put some like an interesting picture on this, but there are cancer types that are more prevalent in younger populations, specifically skin cancer and colorectal cancer. You might have heard that the incidence of colorectal cancer in people under 55 has more than doubled in the last 6 years. So some of the youngest folks who've been diagnosed with colorectal cancer have been 18. So it is like a serious risk factor for young crowds. Part of the reason that doctors think this is occurring is likely due to like dietary influence, reduced fiber. There's a strong correlation between increased fiber intake and lower risk of colorectal cancer. For skin cancer, one of the main contributors is UV radiation, and tanning beds are really, really, really bad in terms of cancer risk for young people. In fact, tanning bed use prior to age 35 increased the risk of carcinogenic melanoma by 75%. So some really scary statistics here. If you want to talk more about those or you want specific links to papers, let me know and I'm happy to provide those. But essentially the idea here is cancer is problematic for human health perspectives and it varies based on kind of which population you're talking about. So again, if you have more questions on that, let me know. Now, this is kind of interesting. I found this figure the last couple of days. This is showing the rate of new cancers in the United States. Again, this is from 2021. But we can see here, blue is more cases per 100,000 people. And then like lighter blue, lighter gray is fewer cases. And this is for all types of cancer across all races, across all ages, ethnicities, and genders. So what are you kind of noticing here when you look at this figure? This is, of course, a map of the United States. But what can we kind of observe here? Eastern, what about the Eastern? We're seeing higher incidence of cancer diagnoses in the Eastern United States. Yeah, I agree. Does everyone else think that seems kind of valid here? Maybe like the Eastern or maybe even like the Midwest, we're seeing a pretty high concentration here. Yeah, so I have a question. Develop a hypothesis for this observation. I want you to think, what the heck is happening here? And I want you to come up with a hypothesis. So our question is like, why are we seeing greater cancer rates in the Eastern United States? And I want you to come up with a hypothesis that answers that question. And it's okay, you don't have all the data, but I want you to think, what is a hypothesis here for this observation in question? Why do we see greater cancer rates in the Eastern United States? That's our question. Does anyone have any thoughts here? Do you need more time? What are you thinking? What is an explanation for this observation? Because remember, a hypothesis is essentially an explanation to a question, right? So what do you think explains this? Sure. Maybe there's more people. Is that like you're saying, like more people are located in those areas? That's a good thought. That's definitely a good idea. So the way that they've developed this figure, it is scaled. So this is rates based off of 100,000 people. So it is scaled for the population. And if we think about places that are the most populous in the United States, California and Texas are hugely populous. New York, of course, also has a really large number of people, but we're seeing very different rates of cancer diagnoses there. So that's a really good thought. Does anyone have any other thoughts here? Why might we be seeing this? Yeah. Absolutely. That's a great thought. So maybe we're seeing greater incidence of cancer in some of these areas due to environmental pollution. Yeah, that's a great hypothesis. And I don't have like the answers to this. This is like an active area of research, but that's a great hypothesis. We know that generally like the Eastern United States had more factories earlier on in terms of like the history of the United States, right? Like the West was developed more recently. So maybe we have more factories. Maybe in some of these states, we have more kind of lackadaisical legislation in terms of pollution. Maybe we have fewer restrictions. As many of you might have seen on like packaging and stuff, sometimes it says like, according to state of California law, this might give you cancer, right? So they have really strict environmental regulations, but sometimes policy and, local governance can kind of dictate that. Does anyone have any other thoughts? Interesting. That's a really interesting idea. Imported foods come from different areas. So people in these places might be getting their food from kind of different areas. Yeah, that's a great hypothesis. Love that. Totally possible. Does anyone think about like what about lifestyle factors or other maybe potential comorbidities? Generally, in some of these areas, there might be reduced access. We might see lower annual income. In some of these areas, particularly like in some Midwestern states, they tend to be poorer than other states in the West, which can reduce access to certain healthy foods and things like that. Potentially, that's what I think maybe. My last question here, what additional data might we want or need? And I want you to talk about this with someone. If we wanted to kind of dig in deeper, what other data do you think would be interesting to kind of like answer this question? What might we need here? So this would be like new diagnoses. So like someone goes to the doctor for the first time, they find out they have cancer. It's not for someone who's like in remission or like undergoing treatment. Good question. Okay, and this is good practice for thinking like a scientist. So, what other data do you guys think we might want or need? Sure, yeah. Population. So in terms of like, are these big cities or small? Is that what you're talking about? Or like number of people in the state? Mm-hmm. So we could look at maybe like, is there a relationship between areas with fewer people or more people? Yeah, absolutely. Maybe we want population data. Yeah, what else? Sure. Yeah, okay, demographic info, absolutely. A lot of times that can be really important for looking at like public health data. A lot of times we see correlations between certain demography or certain demographic characteristics and health. Absolutely. Anything else? Did you have one? Yeah, average income would be super important. Totally agree. Yeah, that could be very interesting to look at as well. And just by the way, these little squares are showing the values for these states that are kind of potentially too small to read on the map. So that's what those are showing. Cool. So this is a great thought exercise. So y'all crushed that. We're going to keep going. Like I said, I don't have answers to this. This is an active area of cancer research. So people are looking at why we see this disparity across the United States in cancer diagnoses. So this data is publicly available. You can go check it out on the CDC website. Going a little bit more into the biology here for the last 10 minutes. Cancer, as an official definition, is basically a collective name for a variety of different diseases that are caused by uncontrolled cell division. Now, typically, this uncontrolled cell growth is a result of genetic mutation, so damage to the DNA that causes proteins not to work properly. And we also see issues as a result of those mutations with the cell cycle functioning. So we have uncontrolled cell growth most of the time because this normal cell apoptosis system is not working. So in a healthy cell, if a cell becomes damaged, it will usually trigger what we call apoptosis, which is sometimes referred to as cell suicide, because it's basically saying like, hey, immune system, come get me. I'm like, no good. I'm damaged. You should come get me. And when it's working properly, the immune system can see that cell, target that cell, and kill it. So then we don't get into trouble with uncontrolled cell growth with damaged cells. However, sometimes cells are able to actually evade the immune system, and that is why we might see cancer, for example. If that first mutation or that cell with a mutation is able to continue to reproduce, that's when we get this uncontrolled cell growth, and occasionally we get cancer. Now, the number of mutations that cancer cells have varies. It can be as few as like 4 mutations. Commonly, it's 4 to 10, but it can be up to like 60 to 100, couple hundred mutations. So questions here on kind of like our basic definition of what we're going to call cancer. Okay. And when we're thinking about these checkpoints, there's 3 important ones. There's others, but these are like 3 main important checkpoints for kind of thinking about cancer. Our first is this G1 checkpoint. So you can see it down here. This is our G1 checkpoint, and this is before the S phase. So before we have our DNA being replicated, most of the time the cell is checking to ensure there's not damage to the existing DNA, because if the DNA is damaged, we definitely don't want to duplicate it and then send it into other future cells, right? So that's a huge point here. G2 is going to be before mitosis. So most of the time, this cell checkpoint is making sure that the cell's big enough, that it has all the nutrients it needs in order to complete that division. They're also going to make sure that the replicated DNA is all good, no problems. So checking for DNA damage is a big portion of these cell checkpoints. Now, if there is damage after DNA replication, so if the cell detects damage at that G2 checkpoint, there actually are ways for cells to repair DNA, and typically the cell will try to do that first. If that's not possible, it will trigger that apoptosis. One of the last checkpoints we're going to talk about is the M checkpoint. This is during mitosis, and this is making sure the spindles are correctly attached to the sister chromatids. So as I was saying, if those spindles are not properly attached, we're not going to get the essentially chromosome arrangement that we want, right? Because if they're not able to pull the sister chromatids apart, maybe we get a duplicated chromosome in one cell and no DNA in the other. That would be a big problem, because we want our daughter cells to each get one of those sister chromatids for mitosis. So the M checkpoint happens actually at the end of metaphase. So remember, metaphase is where those duplicated chromosomes are lining up along the middle, and then anaphase is where they actually split. So the M checkpoint happens right before that split. Because once the sister chromatids are separated during anaphase, that's actually irreversible. The cell can't really like do anything to fix it once they've been pulled apart. Questions here? There are a couple of categories of molecules that contribute to this process as well. These are essentially like proteins that dictate whether or not the cell can go into the next stage. We have positive regulators, which promote the progression into the next cell phase. And we have negative regulators. So when we think about what that kind of means, regulators, of course, are kind of managing, then positive and negative, either like encouraging it or kind of discouraging it. So negative regulators halt that process. And these are acting in addition to those checkpoints. So there's multiple levels of like cellular checks to ensure that the cell cycle stays regulated. We really want a regulated functioning cell cycle. So first, sorry, one more time. If the cell continues like within the cell cycle, like if it's going to go into the next phase. So if it's in G1, for example, these molecules have to be accordingly turned on for the positive regulators and turned off for the negative ones in order to go into the next phase. Does that make sense? Yeah, that bullet is kind of confusingly worded. Regulating if the cell continues through its normal progression. So again, just to reiterate here, positive regulators need to be turned on and negative regulators need to be turned off. Sound good? Cool. I have this break here for you. I want you to be able to do this, but we're only going to spend 30 seconds so we can get back to our cyclins. So share with the partner what you're doing for spring break. Hey, we're gonna keep going. So I heard some fun things. If you want to share with me after, you can. I always like to know what y'all are up to. So honestly, this slide has a lot going on, but don't get too stressed out. So there's a couple important molecules. We refer to these as cyclin-dependent kinases and cyclins. These are basically proteins that help regulate, they promote the cell cycle. A lot of times, if you're reading about cancer biology, you'll see these referred to as CDKs. I actually was listening to a podcast this morning and they were talking about an ad for a cancer research center and they were referring to CDKs just like on the news. So if you've ever come across that, that's what they're talking about here. These are basically like protein kinase complexes. So they're kind of like a combination of different molecules, which is what's being represented here. We see this combination of molecules that basically is associated with activating the cellular cycle. Now, there's a wide variety of different cyclins, and that's what this figure I have here for you is showing. We have different cyclins that are more active at different portions of the cell cycle. And in order for these cyclins to be activated, they have to be bound to these kinases. So the cyclins and the CDKs have to be bound together with the kinase. So again, there's a lot of detail you could get into here. And if you want to read about it, definitely let me know. But this is like a pretty complicated area of like cancer biology. So there's a lot of different cyclins. Something else that's kind of interesting, most of the time there will be cyclins or kinases present in the cells and they need to be activated at certain times. And this activation process is what's being shown in this figure. So most of the time the molecules that are necessary for the cell cycle are kind of just hanging out and then they can be activated. Now this figure is showing like regulator molecule activation. So I'm not going to read this figure to you, but I do encourage you to kind of look at it. You can see associated CDKs and cyclins at different times in the cell cycle. So I'll admit this is a little bit blurry, but it looks better on your computer screen. We can see here we have the different phases of the cell cycle associated with different portions of the CDK cyclin process. So I recommend you check this out kind of on your own time. And I also wanted to mention that there's other molecules like growth factors that influence this transition as well. So I previously defined growth factors, but I didn't put the bullet up here for you. These are just proteins that stimulate cell division. So again, cell cycle is regulated by a lot. There's a lot going on here, honestly. Now, typically regulator molecules are going to accumulate in the cell at different points in time. They drive movement from one phase to the next. So we see this kind of combination of regulator molecules. We also have negative regulator proteins, and those are proteins that stop the cell cycle. There's a lot of really common negative regulators. Those are like RB protein, which is the retinoblastoma, P53 and P21. Same deal, if you're wading through the cancer biology literature, you're very likely to come across some of these terms because they're important molecules for stopping uncontrolled cell growth. We refer to these as tumor suppressor proteins. And most of what we actually know about these tumor suppressor proteins is actually from studying when they were not present. So when that uncontrolled cell growth occurred, so when cancer occurred. Now I have some examples here, and for the sake of time, I'm not going to read them. I'm not going to like test you on these examples, but you can check them out. So read through these bullets a little bit later. As I was saying, cancer is the loss of cell cycle control. We were already talking about cells being able to evade signals for apoptosis. The mechanisms by which they do this are very varied. There's lots of different mechanisms for how they evade targeting by the immune system. So again, lots of papers about that. I could send you some if you'd like, but there's a variety of mechanisms. Now, what's really interesting, and I am going to finish this slide really quick, in culture, which basically means like in a gel media, cancer cells actually don't stop growing. They're defined by like their continued ability to be able to just keep growing, which is why we refer to them essentially as immortal, because when you give them a continuous supply of nutrients, cancer cells can pretty much grow forever. And in fact, we actually have a culture of cancer cells that have been growing since 1951, and they're still growing, and we still use them for biology today. And that's crazy because normally cells only divide 20 or 50 times. So cancer cells are crazy. You should read this book over spring break. It's about these cells. That's it for today. But if you want to talk more, let me know. I can talk to you about any of this. We can chat. Have a good rest of your day. I'll be in my office today, so feel free to stop by. This is my plug for the book, The Immortal Life of Henrietta Lacks. It's a great book. You should all read it. It's very interesting.
The cancer patient that they didn't ask her if they could make her cancer.
Yes, I was trying to get there, but I yapped too much, too long. Yes.